Alrighty. In today's video, we're going to describe. First off, we'll start with the chemoreception of smell and taste, and describe how we get action potentials from eating food. So, we're going to start off with taste. So when we eat something, right, we chew it up, we break it down, and it dissolves into specific ions, right? And we have these grooves on our tongue called lingual papillae, and connected to or close to the lingual papillae are our taste buds. And we have a couple different types of taste buds. This first one is specifically for salt. Second one is for sweet and bitter. Third one is sour. So basically what happens, right? Salt consists of sodium or potassium. Sodium will enter the cell, which will depolarize the cell, which then will open a calcium gated channel, which I don't have drawn, I'm sorry. There'll be a calcium gated channel here and here. Calcium will rush in. Calcium then binds to vesicles, correct? And hopefully you understand this because I have made other videos in the past, but calcium binds to snare proteins that are on the vesicles and on the membrane, which brings it close to the membrane and performs exocytosis. Sweet and bitter on the other hand, is a G-coupled protein receptor. G-protein coupled receptor. So a ligand will come in, it will bind, will produce a signal, which is basically kind of, I guess I could be a little more specific in this, is they have a couple subunits. Alpha subunit then reacts with PLC, phospholipase C, which then releases IP3, IP3 then reacts with the sarcoplasmic reticulum, or endoplasmic reticulum, which releases calcium, right? Got some depolarization going on. Calcium also from the extracellular space will come in. Got tons of calcium. Calcium then binds to vesicles, releasing neurotransmitter. Same thing here with sour. So we got hydrogen ions. Hydrogen ions will come in. Well, sorry. Oh, my Lanta. Sorry. Hydrogen ions will bind and are also G-coupled protein receptors. And we'll do the same process as we just went over. Calcium comes in. Calcium, binds to receptor, releases it. Super simple. And we gotta remember that when the whole G protein coupled receptor is that when calcium is released, it depolarizes the cell, which then allows us to open this calcium channel. So that's taste in a nutshell. Smell. Sorry, I don't have this drawn out beforehand. Oh my goodness. Just trying to draw a nose. That's a fat nose, but we're gonna go with it. So basically what happens is that right here is we have a bone. This is the cribriform plate. And right above the cribriform plate is called the olfactory bulb. And we have neurons that come through here. Okay. And a mucous membrane here. When we smell things, that odorant is going to come in contact with this mucous membrane, dissolve, and then bind here. Well, it's going to bind, basically depolarizing cell basically it just causes depolarization. We could go through it, but it's literally just the same thing as everything else that we've been taught about action potentials. Action potentials goes through the neuron and then goes straight to the brain. That's just a super simplified overview of smell. Now we're going to take a little bit more, take a deeper look into eyes. So what I've drawn out here is a lens. Maybe I'll draw, so this is, okay, we're looking at the eye here from a side angle. I'll make sure we're recording, okay. This right here is the lens. And right here, those are suspensory ligaments, and this is ciliary muscle. And this is going to go all the way around the eye and the lens. Okay. Now, what happens is that I'll talk about this later, and it'll make more sense. But basically, ciliary muscle contracts and relaxes, which changes the shape of the lens. Okay. So if you look at this image, if the ciliary muscle contracts, then it's going to loosen the suspensory ligaments, because it's going to go it's going to contract, and it'll loosen these suspensory ligaments, which will then make more of a circular shape. Now, but if it relaxes, goes out, it's going to tighten or pull on the suspensory ligaments, which is going to create this convex looking lens. Okay, if that, I don't know, take a look at that, see if hopefully that makes sense. We'll hit that just in a second, but here we go. Here we have the eye. We were just talking about this portion here. Okay, now when light comes in from far distance, it's gonna come in, it's gonna hit the iris. Once it hits the iris, it's going to come to this point, and we're going to do some refracting. Now, since it's from a far distance, we don't have to do a ton of refracting in order to hit the focal point or the fovea. Boom. Now, for for like nearsighted objects, we're actually going to have to do a sharper angle in order to hit the fovea. Just because if we don't refract the light at a sharper angle, then we're not going to hit the fovea and we're not going to have a clear image. Now, when we're talking about issues with our eyesight, if I can't see far, right, if I'm nearsighted, and if I can't see things far, I'm going to use a concave lens. So now a concave lens is going to be like this guy down here, this guy. Concave lenses are going to refract the light a lot more so that it can reach the fovea. See, if I used a convex lens if I was like trying to look at something close, it wouldn't do anything. It would refract so much that it would probably end up crossing over. This isn't over exaggeration, but I'd probably end up crossing over like that, and I wouldn't be able to reach the fovea, and it would be a really blurry image. Right? Hopefully that makes sense to you. But on the other hand, if I was looking at something close and it was blurry, right, and I was farsighted, I was looking at something close and it was blurry, I'm going to want to use a convex lens. A convex lens will shorten the distance of refraction, as I've drawn out here in this image. It'll shorten that distance so that I can reach the fovea and have a clear image. Let's see here. Basically, I guess we can talk a little bit about the anatomy. light's going to come in. We'll talk about this right here in a second, but basically that's what allows us to have an active potential, which then goes to optic disc and then goes to the brain and helps us figure out sight. And we have two different types of receptors. We have cones and we have rods. Cones allows us to see color. We have 6 million of those, and rods are what allow us to see black and white. And there are 120 million of those guys, which is kind of crazy. Okay. So in the last image we were just talking, we have basically this is what happens when light hits our eye and allows us to see. This first portion is called a photoreceptor. And then we have our bipolar cell. And then we have our ganglia. Now I'm going to show you what happens when I'm going to show you what happens when light hits this. Yeah, that's what I'm going to do. So we're looking at an on-center cell. Light, so right here, I'm looking at this this protein right here. It's called GPCR, and it's called rhodopsin. Okay, it has a molecule in it called 11-cis-retinol. 11-cis-retinol, right? It's in a cis conformation. So when light comes in, it's going to hit that double bond, and it's going to do a conformational change, and it's going to shift it to trans. That shifting is going to kick out. It's going to come in. It's going to kick out that molecule, just like so. When it does that, it's going to release transducin, or this alpha particle. This alpha particle is going to react with PDE, which is phosphodiesterase. Phosphodiesterase will react with cyclic GMP, which this PDE is going to actually destroy cyclic GMP. Just gotta remember that. We'll go over this again later. This sodium channel is going to be open. No, it's closed. So sorry. Okay, so essentially, right, if this sodium channel was open, then we could have an influx of positive, and then we'd have an action potential. But that's not the case with this light. Okay, so nothing's released. Nothing is released into the cleft, but then we get to this point, and we have another protein, mGluR6. mGluR6 is a ligand channel, and it's going to react with glutamate, but there's no glutamate in the cleft right now. So that means this calcium or trypsin-1 channel is going to stay open, and it's going to allow for calcium to come in and allowing an action potential. The action potential will then open this voltage gated calcium, and it'll bind to the vesicles, pulling it towards the membrane, releasing glutamate into the synaptic cleft. Glutamate will then bind to AMPA, which is a cation channel or just sodium channel. Okay, which then allows for the influx of sodium, creating an action potential to the brain. Okay, that's with light. Hopefully you followed. If not, that's fine. Now we're gonna just go over, see what happens when we're basically looking at something dark and there's no light hitting our eye, these photoreceptors. So we won't have a conformational change. And this 11-cis-retinol is just going to come through. It's just going to come through. Nothing's really going to happen. So if that's the case, then this this alpha receptor is not going to activate PDE. So if it doesn't activate PDE, then cyclic GMP is not going to be deactivated. So if cyclic GMP isn't deactivated, it's going to act as a ligand. It's going to attach to the sodium, which is going to open the channel, allowing for sodium to enter. It's going to create an action potential, open this voltage-gated calcium channel, and then, right, calcium allows for glutamate to be released, and then the glutamate is going to bind to the mGluR6. mGluR6 going to release an alpha particle. Alpha particle is going to bind to trypsin-1, which will deactivate trypsin-1 and close the channel. Close the channel. Channel's closed. No action potential occurs. So that means no glutamate is released. And that means that no action potential can occur. So that is kind of hopefully that made sense. I want to just take a look, before we jump back up there, I want to take a look at receptor fields, kind of how those work. So in our fovea, we have a ton of these guys. We have a ton of on-center, a ton of on-center receptors. But on the outside too, as in seeing this image, we also have other, basically off-center. And so off-center is going to act a little bit different. Off-center will produce an inhibitory response to my on-center. So we're just going to take a look here. Remember we have we have an on-center receptor. On-center is going to it won't release glutamate, right? It's not going to release glutamate. And we'll get to the synaptic cleft. Sorry. Maybe I'll use this image maybe a little bit better. Yeah, okay. That's a little bit better. Okay, give me 1 second. Let's go. Okay. Okay, I understand. Okay, there's a difference. I'll talk about it right here. I'm really sorry I'm kind of jumping everywhere. On-center photoreceptors react to light and produce action potentials through GluR6. Off-centers will react to dark and will also produce an action potential through the AMPA channel. Here we'll look at an off-center photoreceptor, right? No light, the light doesn't do anything, right? It doesn't change this 11-cis retinol, so that means that GCMP can still be activated, allowing for action potential to come through, releases glutamate. But then on this bipolar cell, it's going to bind to an AMPA channel right here. AMPA channels, it'll bind, sodium's going to rush in, create an action potential, release more glutamate. It's going to bind, create action potential, okay? Now, if this was an off-center, well, maybe just say off-surround. I'll say that. This is an on center. Right, the on center, as we remember before, it's going to react to conformational change. This guy's going to come in, react with that. It's going to deactivate this guy, means this guy stay close, so no action potential occurs. But off surround, light hits it, we'll have glutamate released, glutamate binds, then it releases GABA or of some sort, and which then hyperpolarizes the cell. Hyperpolarizes the cell, which then allows for a Buang to happen. Even though we're in the dark, we can still have an action potential occur because of the horizontal cells. Hopefully that made sense. Now we're going to take a look here and talk about the ear. Okay, so we'll do a little bit of anatomy first before we get into this. Sound waves are going to come in through the ear. It's going to come in contact with the three bones, malleus, incus, and stapes. This is called the middle ear. Those are going to amplify the sound, which is again going to come in contact with the cochlea. The cochlea has three different parts: the scala tympani, the cochlear duct, or the scala media, and the scala vestibuli. Okay. In the scala tympani, there's a basilar membrane. The basilar membrane it reacts to the sound waves coming through. Okay. And depending on the frequency, this section will be high frequency, this will be lower frequency. Just remember that plays a really big role in how we hear sound. So now I just want you to know, I made a transverse cut there, and now we're looking at it head on. I apologize, there's a lot going on, but I'm going to explain really quick. Basically, in this area called the stria vascularis, right here, we have these pumps called NKCC, which basically is just transporting a bunch of potassium into the endolymph. So we have a ton of potassium coming into the endolymph. Here. Tons of potassium coming into the endolymph. Tons of potassium, I mean a lot. Okay? Just remember that. Now we're going to take a look here at the hair follicle. Hair follicle consists of 3 fingers, and on those fingers are voltage-gated potassium channels. Now if these fingers shift, it's going to open the gate here to allow for potassium to come in, which then would essentially depolarize the cell, open the voltage-gated potassium or calcium channel, and allow the action potential to occur. So this is what happens. You hear a sound, right? If it begins to resonate with the basilar membrane. The basilar membrane is right here, and it starts to resonate. It starts moving up and down. That makes these hair cells push in or stick into the tectoral membrane. Now, right when we were talking, if those hair cells come in contact with the tectoral membrane, it's going to shift those hair follicles, right, and allow for sodium to enter in. Potassium. potassium's going to rush in, okay, because of the crazy driving force that it has. Potassium's going to rush in. I need to look at the hair follicle. Okay, potassium's rushing in. It's depolarizing the cell. Okay, we also need to remember that in the cell, hair cells, we have what's called prestin. Prestin is basically a protein that's going to contract and relax the hair follicle. So it's going to make it go up and down. It's going to cause this due to a change in voltage, right? And so since the potassium comes in, it's going to change the voltage and activate prestin, which is going to actually resonate more. So now we have the basal membrane going, and now we have prestin going. And this movement, this up and down movement, it's gonna bring the tectoral membrane completely down to the inner hair follicles. And the same thing's gonna occur, but this is the important part. If you have glutamate, or not, a neurotransmitter released here, nothing really happens. You don't really necessarily know, but it's not going to the brain. It's not going to the brain. Okay, we're just telling the cell, we're just moving that basilar membrane and tectoral membrane enough so that we can hit this inner hair cell. Once the inner hair cell is hit, potassium will rush in, and the same thing will happen. We'll have an action potential. Then this will actually release neurotransmitters that will go to the brain, right? To the brain, telling us what we're hearing. These external hair cells actually are efferent pathway. It's an efferent pathway, so it's coming from the brain, and this is kind of how we block out noises. If we're in a crowded room, right, and there's a ton of noise, our brain can actually send signals back to these hair cells, telling them not to move as much. And in that case, if they're not moving as much, they're not there's not much resonance, right? And so this tectoral membrane isn't going to reach that inner hair cell, which will then mean less action potential from the inner hair cell going to the brain. Yeah, hopefully that all made sense. I did kind of rush through it pretty quick, but yeah, that's that for you.